Good morning, good afternoon, good evening, everybody. It's a pleasure for me to be here today and introduce to you the 1 hour automated mycoplasma test for cell therapy products using a innovative way to perform molecular tests using what we call a lab in a pouch system. And I think that this technology match well with the subject of the webinar today, that is leveraging instrumentations to accelerate release testing and minimize human inputs. And before to start and to going into the technical details, I would like to tell you what the topics I want to cover today. So give you a little bit of background on mycoplasma testing, what is the BioFire mycoplasma solutions we develop, different sample preparations, then raise some conclusions about the use of the technology, and we can move forward later to the Q&A sessions with the other participants to this webinar. So, as you probably know, mycoplasma contaminations are obviously rare events, but however, when this can occurs, it's a bit messy, right? So it generates a lot of issues. Independently of the fact that obviously a mycoplasma contaminations can alter the property of the cells that we are using, either for bioproduction or cell and gene therapy products. And most of the time, if this is not detected, we can release products that are unsafe for patients. In addition, the testing of mycoplasma present or the contamination of mycoplasma in development and manufacturing is a requirement by worldwide regulatory agencies. So we need to be compliant with these regulations, and we need to test it. And in addition, the analytical methods for mycoplasma detection are already described in the major pharmacopoeias, the United States Pharmacopoeia, through the general chapter number 63, but also the European Pharmacopoeia chapter 2.67. That, by the way, this chapter is under revision today, and there are some upcoming additional requirements included in this chapter, like, for example, the requirements to replace culture methods and cells, and the cells should be included as part of the testing materials, and not only the supernatants. And the last pharmacopoeia also including the describing how the mycoplasmas are detected is the Japanese pharmacopoeia. So extremely regulatory and compliance link of the mycoplasma detection here. However, did you know that 95% of cell culture contamination is made up by only 6 species of mycoplasma? And these species are Mycoplasma orale, Mycoplasma fermentans, Mycoplasma urinis, Mycoplasma hominis, Mycoplasma arginini, and also Aculoplasma laidlawi. And also the most common mycoplasma that contaminates cell culture originate from human, bovine, and swine. And also up to 1,000 mycoplasma cells can be attached to an infected cells at any given time. So, and I think that this is an important parameter because some of these mycoplasma can be present but cannot be detected eventually in the supernatant because some of them could be intracellular, or some can be attached to the surface of the cells. So meaning that this is also one of the reasons the European Pharmacopoeia is including to test the cells during the mycoplasma testing and not only the supernatant. So the current mycoplasma testings methods are 2 types. The first one is what we call the compendial methods, and there are 2 different ones. It's what we call the cell culture methods, in which we will incubate the samples in a specific culture media and wait for 28 days the appearance of the colonies. And this is, you can imagine, arduous, require technical expertise, and it's taken so long time to get the results. The second one is what we call the indicator cell culture. And in this case, the sample is incubated in the presence of cells, there are cells, for example, that becomes contaminated, and then we look at the presence of the mycoplasma intracellularly. And obviously, the limit of detection for these two methods is different. The first one is requires 410 CFUs, and the indicator cell culture, the LOD expected is 100 CFUs. And the second method that and is the most common one employed today is what we call the alternative method. So it's a PCR-based method, and this is more rapid time to results because we can get results in less than 5 hours today. But again, this requires some level of expertise of the operators running these assays, and need some specialized lab to avoid any contamination with amplicons or other mycoplasma, etc. And as mentioned, it's prone to contamination. So we need to find a way to do how we can improve this mycoplasma testing in a routine base in a GMP compliant laboratory. So the other point, and based on the topic today, is how to reduce human errors, right, in human mycoplasma. And before going to that, so we need to see what are the most common human errors when we run a PCR testing. And this could be general, but also including mycoplasma. And different factors has been listed, like the technician experience, following the right SOPs, the calibration of the equipment, and also the procedures for the quality control of the lab. And reading some publications, the estimated suggested human error for the PCR process can range between 1 to 5% when a lab is well controlled in a well environmental setting, but this could be to 15% when the PCR is performed in less controlled environment. So this is pretty high, because every time that we have a negative result or invalid results implies investigations that mobilize a lot of people, and trying to minimize that is one of the goals we have been trying to do. And the human source of human errors also in PCR for mycoplasma testing can come from contamination of the sample, some pipetting mistakes, and the previous talk inspired that, so the incorrect reagent preparation, some programming errors during the thermocycline preparation, and so on. And the automation and stringent quality controls also help to reduce these errors, right? And has been demonstrated that some of these automations can push the error rates below 1% in high quality labs. So automation and reducing human errors is critical for the mycoplasma detection. So let me go now to explain to you what is the BioFire mycoplasma solutions that we are proposing in bioMérieux. So this is in this slide you can see the system. So the film array, the BioFire pouch, and all the ancillary reagents that are needed to run the assay. So it's an automated closed system to detect presence absence of more than 130 mycoplasma species. It's a molecular lab in a pouch, because everything that is needed to run a molecular assay is included in the pouch already, and contains all the reagents needed for the PCR. Have a simplified workflow with minimum hands-on times. We still need some people setting up the assay, but it's very simple to do. The results are provided in 1 hour, and the software is a very intuitive, is given results in terms of mycoplasma detected or mycoplasma not detected. And you will see that we have also internal controls that allows to validate the assay at the end. And the software is 21 CFR Part 11 compliance, ensuring all the data integrity that are needed in the pharmaceutical industry. And this is a very short chart how the assays work. So you have the pouch, you put the slide into what we call the loading station. So we use 2 vials, one to rehydrate all the reagents, the second one to put the sample. And then once the pouch is loaded, is put in the instruments. we have a barcode reader, so we can trace everything that has been happened, and trace also and link the samples to the pouch and to the product. So the BioFire rapid PCR technology is composed by two different assets here. The first one on the left-hand side of the slide is the film array instrument, and it's pretty small instrument. It's like a kitchen bench instrument. And the second one is what we call the BioFire pouch. Okay, and this is automated and it's a closed system. And in the pouch, you run and you can have all the different steps to run this assay, from the extraction of the nucleic acids, amplification, and detection. So all in one in a very small footprint here. So let me tell you how this molecular lab in a pouch work. So obviously here you see is one pouch is one test. The pouch is stored at room temperature, so you don't need to have any cold rooms for this. And on the top of the pouch, you have all the freeze-dried reagents here. And then you have the what we call the fluidic circuits, where all the liquids will be moved from one chamber to another using some actuators in the system to go into the array that will perform the single assay. So the first step is the sample extraction and preparation. So the samples goes into this gray area here. We have a beating sample extraction. So the cells are destroyed in order to release all the nucleic acids in the sample. And then we use some magnetic beads for nucleic acid purification and release. Okay, so with the assay works with mRNAs and DNAs. So meaning that because of we are also running and detecting the mRNA that is related to the live mycoplasma, and we know that the mRNA is highly expressed in mycoplasma in function of the strains, thousands of copies of this. And we perform the reverse transcription, and then we perform the first multiplex PCR. Okay, so different primers that are used to amplify different genes. In that case, we use 3 genes are target that allows to detect the 130 species of mycoplasma. After the first PCR, the product is dilute and distribute in the array, in which we have 14 specific PCRs that are performed in triplicate. And this second stage PCR will amplify the inside of the original amplicon that has been amplified from the first PCR in order to increase the sensitivity and really identify what are the specific genes related to these different mycoplasmas. Okay, so and each well contains a specific set of primers for an individual PCR. So we know that only the primers that has been set in the well will be amplified only a specific fragment from the pool of amplicons that has been generated during the PCR1. In addition, so we have a fully integrated process control. So eliminating all these tedious manual steps in which you add manually all the different controls. So we have a freeze-dried yeast at the beginning that is part of the full assay, allows to nucleic acid extraction, allows to check the reverse transcription, the PCR1 and the PCR2, right? So and also we we develop some specific what we call external positive controls that allows to do the quality control of the incoming kit, of the incoming QC, or it can be used for the training of the operators, but also for the qualifications. And something that is important and we take into account here is these synthetic modified mycoplasma DNAs has been modified in order to shift the temperature curve of the original amplicons coming from the live mycoplasma. So in order to avoid any contamination mistakes when the sample is tested, right? So there is no possibility to mismatch or to contaminate a sample with this external positive control. I have results that are not corresponding to the sample. So when we compare the BioFire method versus the conventional nucleic acid testing, so you see this is a conventional PCR method. You have your sample, you have the extraction controls, you have different instruments that are used. So the nucleic acid extraction system or the reagents, pipetting it, or using some automates also in a 96-well microtube plates. This goes into the real-time PCR, and then you have all this data analysis that needs to be performed. And this can take 5 to 7 hours. With the BioFire mycoplasma, this takes less than 1 hour because everything that you can see on the top of the slide is only included in the pouch and at the same time in the thermocycler. So you have all the different steps on nucleic acid purification, reverse transcription, multiplex PCR, and analyte detection, and automated result analysis that is performed by the system. And before to validate the assay, obviously all the internal controls needs to pass the specifications that has been predefined in the instrument. So we validate the method following all the international pharmacopoeia standards and all the everything that has been written about the validation of the alternative methods, meeting the expected LODs coming from the different European and USP and Japanese pharmacopoeias. The LOD must be less than 10 CFU per ml to replace the culture method, and the LOD must be less than 100 CFU per ml to replace the cell culture method, right? Based on the European pharmacopoeia. And because this is a qualitative test, so we needed to validate some parameters that are described and are requested by the different general chapters or technical documents, like the specificity, the detection limit, the robustness, the repeatability, the roughness, and also do some equivalency studies versus the compendial methods. So in terms of sample preparation, we validate different protocols today that allows to provide some flexibility in the terms of the use of the technology. And we have four protocols. One is in which 10 ml can be analyzed, 10 ml of supernatant using a double centrifugation. The hands-on times is 25 minutes. The LOD is less than 10. The type of samples used is supernatants. The second one is another protocol in which we can use cell-containing samples. Is a derived protocol of the first one. It's also taking 25 minutes less than 10. And we developed recently, and this is what I would plan to explain to you today, is the 1.7 ml double centrifugation protocol, 25 minutes hands-on time. Also, we reach the limit of detection that is required by the regulators. And in that case, we can use cell-containing samples. And this protocol is perfect, for example, for the CAR T-cell products. And we have also a protocol, what we call the in-process protocol, in which you can spike directly 200 microliters of the supernatant in the pouch. This takes 2 minutes, but the LOD is less than 33 CFUs per ml. And this is because one of the strains are detected at more than 10 CFUs, right? Most of them are detected at 10, but some are 30. So we decided to set the cutoff to 33. And in the case when we talk about microbiology for in-process testing, 10 or 30, it doesn't make sense, right? So it's pretty much the same. But from the regulatory standpoint, we need to have 10 CFUs. But for in-process, this can be very powerful protocol to screen and to run and to look at if some potential contaminations may occur during your process. And this is for the sample supernatants. So the protocol that the data I would like to show you today is from the protocol described on the bottom of the slide, in which you have a CAGT sample containing cells. So we combine the number of cells that the pouch can be managed and the supernatant at the same time in order to have the supernatant and the cells in the sample that will be analyzed by the biofile mycoplasma. So we have a first centrifugation here. We recover a small sample from the pouch. We mix the cells with the supernatant, and then we spin everything. This gets supernatant. We added the sample buffer and then load into the pouch. So pretty simple protocol here. When you look at the data, we use URCAT cells, 10 to 7 cells per ml in all these experiments. That is pretty much in the range of the cell concentration of the CAR T cell products. As you can see here, the different microorganisms that were spiked on the left-hand side of the slide, the stock reference, some of the data of the pre-LOD studies that we perform, and then we spike to confirm the LOD we get it from the different microorganisms, and the number of the replicates we have to give us the detection rate. As you can see here that all the mycoplasma were in the percentage that is expected. And most interestingly, there is no false positive results were observed, okay? And we have the appropriate sensitivity for all the mycoplasma tested, the detection rate of 95% for 24 replicates has the pharmacopoeia requirements. And in terms of inclusivity, so inclusivity is really to see other mycoplasmas can be detected also in the context of the assay, again, including 110 to 7 URCAT cells per ml for this low volume protocol. And we can see here the Mycoplasma bovis, Mycoplasma hominis were detected at the detection rate. And Ureplasma urealyticum is detected at a little bit less than 100 Celsius in that case. And when you look at the exclusivity, so it's really demonstrate that all the closely related bacteria cannot interfere, okay, even at the high concentration of these microorganisms with the sample. And we tested different microorganisms from the Bacilli genera and the Clostridia. And you can see here the concentration test, and the results were negative. So the mycoplasma were not detected, showing the real specificity and exclusivity of the assay for the detection specifically of the mycoplasma. And this nested PCR we use is very key to have these kind of results in terms of specificity and inclusivity. And we tested also a myriad of different products that has been described to interfere with the PCR technologies. And except the bleach at 1%, any of these different cell lines, microorganisms, disinfectants, cryoprotectants, antimicrobials, enzymes, etc., show any interference with the BioFire technology, and we still detect at the LOD the different microorganisms. The robustness was evaluated also using Cheiloplasma lelawea, Mycoplasma urinae, so that the worst cases that has been described in the literature. And we tested 15 variations in the parameters, different parameters in terms of pre-processing steps, sample buffer ratio, different feature of the cell matrices, and all the results were in the expected results, more than 55% for each sample. So the assay is robust for to be performed. And we have now adopters of the BioFire mycoplasma. Some of these has been publicly communicated. Amgen is use the BioFire mycoplasma for CHO-derived products. Johnson & Johnson, they already have some approvals to use the BioFire technology for the release of the products. And you can see here the presentation, the posters that has been, or the presentation that has been done at different international conferences, like the Rapid Microbiological Methods Workshop organized by the PDA, the Pharma Lab, some articles in the European Pharmaceutical Review, some webinars available on demand, and so on. Some different matrices, including, for example, for berry cells on chondrocytes from porcine collagen membranes, Dana-Farber-Cancer Institute, Novartis, and a very nice study performed by the NIH at the laboratory of the Professor Ann Lowe, in which they described that the BioFire technology mycoplasma demonstrated the lowest LOD across all the platforms tested, and also testing the presence of tumor-infiltrated lymphocytes did not impact the sensitivity of the assay. So I would like to conclude with two different slides to show you the benefits of the BioFire mycoplasma for labs using traditional PCR. So obviously we can increase the lab efficiency because we streamline the workflows, we simplify the training and reducing the learning curves, improve data integrity, reduce the subjectivity of human errors and retestings when some invalid results occurs. We haven't seen any of these today in the BioFire mycoplasma. So process optimization also, because reduce the burden of the no quality, reducing the investigations linked to the human errors, less human resource use, the centralized testing, so eliminate requirements for molecular lab for mycoplasma testing here for in-process outline. So we can ask you, what is the impact that you think these efficiency gains will have in your organization here? And the last one is the accessibility of in-house mycoplasma testing, right? So eliminate the costly outsourced testing. This is an easy three-step process. You don't need to have a very specialized molecular lab or expertise required. Response and recover quickly from contaminated events, knowing the results in less than 1 hour. And the most important also, we are create confidence and compliance, Be confident in the method you are used. You will meet the pharmacopoeia requirements, and you have your process under control. And also, so how do you think? Easy to use 1 hour in-house mycoplasma test will impact your organization here. So with that, I would like to thank the teams in bioMérieux, the BioFirst teams that supports all along the development of these protocols, our team in Craponne, in Lombard, and also my colleagues of the Global Marketing Manager, Jeremy, and Julie Decken from the Markham. And you see my email on the bottom of the slide. So if you want to learn more, obviously, I invite you to follow the bioMérieux Pharma Quality Control website. You have plenty of information, plenty of content related to the technology and how this technology is really making a difference in the way the CAR-T products are controlled. Thank you very much.